Assalamu alaikum and welcome everyone to our Molecular Biology ASCP preparation series. This program is all about providing real life MCQs for your certification success. The MCQs we'll discuss today and in upcoming session have been carefully collected from several years of the past exam to give you the best preparation possible. I'm Dr. Lai Abdelati, certified as international technologist in molecular biology, and I'm really excited to share my knowledge and experiences with all of you through this series. I also encourage everyone taking the test to share your question with us so that everyone can benefit. We are continuing with the first part of molecular science, focusing on the key aspect of nucleic acid chemistry. So let's dive in together, but don't forget to subscribe to our channel to receive all the latest lecture. Are you ready? Let's get started. Okay, here is what we'll be covering today. First, we'll go through the remaining MCQs about the sugars, nitrogenous bases, and chemical structures. Okay. Second, we'll dive into associated proteins. And third, we'll focus on the mutations, including their types and effects. So now let's move on the first question for today. Okay, here is the first question. What is the structure of a phosphodiester bond in DNA and RNA? Actually, I have set a timer for 10 seconds, but feel free to pause it if you would like to have more time to read the question slowly, okay? All right, the correct answer is B. Actually, here we describe how the phosphodiester bond connects two sugar molecules within the backbone of DNA and RNA strands, forming a continuous chain from which nucleotides are attached. So the option or correct option here is link the 5' carbon atom of one sugar molecule to the 3' carbon atom of another. All right? Okay, let's move to the next question. Okay, how many nucleotides specify amino acid? All right, 10 seconds. Actually, each amino acid is defined by three nucleotide sequence called the triple codon. All right, these three nucleotides, also known as the codon, determine which amino acid will be incorporating into growing protein during the translation. All right. So the next question is how the formation of phosphodiester bond facilitating during the nucleotide polymerization. Okay, give you 10 seconds. All right. All right. The answer is by breaking apart the triphosphate or diphosphate form of nucleotide to release energy. All right. But let me explain it briefly. Actually, to form DNA or RNA, what will happen? Nucleotides must join together, right? Through the process called polymerization. As we know, nucleotides are the building block of DNA or RNA and consisting of nitrogenous base, sugar, and phosphate group, right? These phosphate group are the key because they help form the phosphodiester bond, the links between nucleotides. So before this happens, nucleotides like ATP need to be activated. This occurs when they release two phosphate group at the pyrophosphate. The energy released from the breaking this bond drive the reaction forward, allowing nucleotides to connect and build the DNA or RNA strand. I hope it's clear, okay? So let's move to the next question. The next question. Which of the following statement about the DNA and RNA is true? All right, the correct answer is C. DNA is more stable than RNA. Why DNA is more stable than RNA? Said DNA is more stable than RNA due to the legs of the 2' hydroxyl group in the deoxyribose, as we said earlier, right? Which make RNA more prone to hydrolysis. Okay, now let's move on to the associated protein MCQ section with the nucleic acid chemistry part. All right. So the first question on this part say DNA and histone interaction is primarily due to, see the option below. Actually, the interaction between a DNA and histones are primarily due to ionic bonds. But let me explain it in simple way. As we know, histones are the positively charged protein found in nucleus of eukaryotic cells. They play a crucial role in packing DNA into a compact organizing structure called chromatin. DNA, on another hand, is negatively charged molecule due to the what? Due to the phosphate groups in the backbone. All right? So, the positive charge of histones come from the basic amino acid like lysine and arginine. This amino acid have positively charged side chain. So, focus. When the DNA wraps around histone, the negatively charged phosphate groups interact with the positively charged amino acid of histones through the what? Through the ionic bonds. This interaction helps stabilize the DNA histone complex. I hope it's clear now. So let's move to the next question. Okay, which amino acids are primarily associated with histones? 10 seconds. All right, now let's go ahead and see the answer. All right, the correct answer is B, lysine and arginine. Actually, together, lysine and arginine contribute to the overall positive charge of histones, as I said earlier, allowing them to bind to the negatively charged DNA and organize it into nucleosome. So the correct option is B. Let's move to the next question. What role do lysine and arginine play in the histone structure and function? I think this is a simple question. The correct answer is B. Actually, lysine and arginine residues in histone undergo various post-translation modification, including acetylation and methylation. So this modification regulate chromatin compaction, gene expression, and other cellular process. So the correct answer is B, is undergo post-translation modification to regulate chromatin structure. Okay, let's move to the next question. Okay, next question say, how do lysine and arginine contribute to chromatin organization? 10 seconds. Okay, the correct answer is by neutralizing DNA charge through the ionic interactions. Okay, actually, when DNA wraps around histones, lysine and arginine residues interact with the negatively charged phosphate group of DNA through the what? Through the ionic bonds. This interaction helps neutralize the overall charge, allowing DNA to be tightly packaged. All right. Okay, now let's move on to the mutation MCQ section with the nucleic acid chemistry part. Let's go. So now before we move on the questions, let's first look at the summary of the four main types of mutations. I suggest pausing to read it before we answer the next set of questions. All right. So now let's take a look at the question that appear on the molecular biology ASCP exam related to the mutations. Here the first question said, which of the following is the example of missense mutation? All right, 10 seconds. All right, let's move to the answer. All right, the correct answer for identifying a missense mutation is option B, UGC to UCC. Actually, a missense mutation occurs when a single nucleotide change in the DNA sequence results in the codon that specify a different amino acid. All right? But before you jump on the next questions, thought take a minute to brush up on the other mutations type we see in the table here. For the nonsense mutation, this one like a stop sign in the middle of the road. It turns a codon into stop codon, cutting the translation short, all right? For the silent mutations, these are the sneaky mutations. A nucleotide changes, but the amino acid stay the same. Okay? And for the frame shift mutation is the big trouble here. Add or delete a nucleotide and the whole reading frame gets thrown off. All right? Just remember these sentences and will not forget or mix between the type of mutation. All right? Let's move to the next question. The question here, what is the name of mutation that changes the reading frame of the RNA molecule? 10 seconds. Okay, let's move. The name of the mutation that changes the reading frame of RNA molecule is called frame shift mutation. This type of mutation is caused by insertion or deletion of the number of nucleotide in DNA sequence. So which alter the messenger RNA reading frame during the translation process. Okay, so the answer is frame shift mutation. Okay, which type of mutation involves the addition of one or more nucleotide base pairs into a DNA sequence? All right, 10 seconds. Let's go. Substitution, deletion, insertion, translocation. Here is the options. Okay, now let's move to see the correct answer. The correct answer is insertion. An insertion mutations occurs when extra nucleotide base pairs are added into a DNA sequence, which can disrupt the normal function of a gene. All right, let's move on. So the next question is, what is the primary consequence of the frame shift mutation? All right, the correct answer is B. It caused a shift in the reading frame of the genetic code. All right. Actually, the frame shift mutations result from the insertion or deletion that are not in multiples of the three nucleotides, causing a shift in the reading frame. This alters the entire downstream amino acid sequence, often leading to non-functional proteins. All right. So now let's move to the next question. All right, the question here is, Which of the following best describe as silent mutations? 10 seconds. A silent mutation happens when a single nucleotide in a DNA sequence is changed, but the resulting codon is still code for the same amino acid. This occurs due to the redundancy in the genetic codes where multiple codons can specify the same amino acid. Let's see the example here. You see the original codon GAA codes for the glutamic acids and the mutated codon GAG also code for the glutamic acids. Since the amino acid doesn't change, there is no effect on the protein structure or function. All right, so let's move to the next question. All right, the question here say, which of the following mutation is the most likely to be neutral, having no effect on the organism? Nonsense mutation, missense mutation, silent mutation, or frame shift mutation? Thinking, 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 and let's move to see the correct answer. All right. Silent mutation. Silent mutation do not change the amino acid sequence of a protein and therefore typically do not affect the protein's function or the organism. All right. Okay. What is the term for a mutation that result in the premature stop codon? All right. Nonsense mutation introduce a premature stop codon into DNA sequence, leading to the production of truncated, usually non-functional protein, as I said earlier. All right. So the answer is nonsense mutation. All right. The question here is mutations in which type of cells can be passed on to offspring. All right. 10 seconds. Somatic cells, gametes, muscle cells, nerve cells. All right. Let's move to the answer. Yes, the correct answer is gametes. Only mutation in gametes, like such as the sperm or egg cells, can be inherited by offsprings, affecting the genetic makeup of the future generation. All right, so the correct answer is gametes. Let's move on. All right, next question. Which type of mutation is caused by the exchange of the segment between non-homologous chromosome? All right, the correct answer is translocation. Actually, translocation mutations involve the exchange of the genetic material between non-homologous chromosomes, okay, which can disrupt the gene function and lead to disease. All right, so the correct answer is translocation. Let's move to see the other question. Okay, the question here is what is the effect of a missense mutation? 10 seconds. All right. As I said earlier, missense mutation result in substitution of one amino acid for another in the protein sequence, which can affect the protein function depending on the location and nature of the substitution. All right. So the correct answer here is it changed one amino acid in the protein sequence. Okay, this is the last question in molecular science part for today. Thank you for your attention. I will see you in the next lecture. So before you go, don't forget to like, comment, share, and subscribe to our channel to receive all the latest lecture. Salam.